Hey guys, it's Professor Tiffany Martin with Medical Microbiology, and we are in Chapter 15, Innate Immunity. And we have been working our way through physical barriers over here with the first line of defense. And then we have now gotten into the second line of defense, and we just finished a video about the different cells. So we're still in the cells section, but I wanted to explain a little bit about how the cells kill. So we're going to talk about, this video talks about phagocytosis, which all of those cells that we talked about, almost all of them are capable of phagocytosis. So we're going to talk about phagocytic ways of killing, and then a couple of mentions about non-phagocytic ways of killing. So phagocytosis, again, this is something you've probably been experienced with. Any cell that is capable of this process of phagocytosis, which is kind of eating up and killing foreign invaders, any of these cells can be called phagocytes. So an eosinophil is a phagocyte, a macrophage is a phagocyte, because these are cells that are capable of phagocytosis. This textbook divides it into kind of 6 parts. If you've learned it a different way, that's fine. If you haven't, let's learn it this way. I always like to, when I'm studying, come up with these well, before we get into that, let's watch this video about phagocytosis, and so we can actually kind of see how this is going to work. Let's see if I can get that on. Okay, so this is a YouTube video about phagocytosis. And we're going to kind of watch this just to kind of get an idea of how it happens. This large cell is our neutrophil. So this is a phagocytic blood cell. We have a red blood cell. And then this is, they're talking about the bacteria that are inside our blood. And we have a neutrophil that is picking it up. Now, neutrophils, the bacteria will produce some chemicals, and the neutrophil will actually chase this down. It's following these chemical signals and looking for this bacteria. So you can see it moving. It moves by pseudopodia, and kind of will track down this bacteria until it can finally ingest it, and then it will kill it and eliminate it and destroy it. So this is the process of phagocytosis. So let's go back to our, oh, that's me. Let's go back to our, okay, so let's go back to our slide with phagocytosis, and let's label the steps that we just watched. All right, so first one is chemotaxis. Then we're going to have adherence, ingestion, maturation, killing, and elimination. So just using the first letters I created with this little mnemonic that helps me remember it. You, of course, can create your own, but I love doing this because it helps my brain process things better, especially when I'm trying to memorize a list of things. So CAIMKE, I put together calling all invaders, must kill, eliminate. So that's my little mnemonic. Calling all invaders, must kill, eliminate, and that's how I don't miss any of these steps. So starting with the C, that's chemotaxis, and we saw that this bacteria is producing chemicals. It's creating a chemotaxis in the phagocyte. Taxis is movement, so the phagocyte is actually moving towards the bacteria, and we said that it moves by pseudopodia. Once it reaches the bacteria, we have to have adherence, calling all invaders. The A is for adherence. We see this with viruses. Things have got to attach. So adherence is very important for this phagocyte to move through and get to the next step. This process is called opsonization, this adherence, this attachment part. Opsonization actually translates into preparing to eat. And we talked about how some pathogens have virulence factors that help them evade detection from the immune system. And we said that if a bacteria has that smooth capsule, it makes it harder for the phagocyte to grab a hold of the bacteria. So that smooth capsule is considered a virulence factor. If a bacteria has that, it's more likely to be a pathogen. So, so getting a hold of that adherence is important. There are some chemicals that we can make, some molecules that we can make, that our body makes, that will attach to pathogens to help a phagocyte to find it and bind on. Those factors are called opsonins. So know this vocabulary term because it's going to come up a couple of times in this as we work through this course. So adherence by the process of opsonization, that attachment and adhering. Opsonins are molecules that adhere to the bacteria cell to enhance opsonization and make that process easier. Once it's adhered, calling all invaders. The I stands for ingestion. It's going to take it into a vesicle called a phagosome. That phagosome is going to fuse with a lysosome, and that starts the process of maturation. The phagosome and the lysosome fuse and become a phagolysosome. The lysosome is something the cell makes, and it has all of these different chemicals in it. These chemicals will kill bacteria. Might be enzymes, might be reactive forms of oxygen, or other things that will kill bacteria. So now we are at the calling all invaders must kill. We're at the killing stage, and then elimination, and this is by exocytosis. If we want to look at the picture, pictures are so much better. And there's my bacteria. Here's my phagocyte. And we can see the pseudopodia as it's moving and wants to adhere to that. So I have chemotaxis and then adherence. It comes inside ingestion. And moves into something, a vesicle called a phagosome. So after ingestion, your book says maturation, and that's the fusing of this phagosome with the lysosome that is produced by the cell and has these chemicals, reactive chemicals, enzymes in it. This makes a phagolysosome, so maturation. Killing occurs inside the cell, and then elimination by exocytosis. Now, I do want to point out that this is an innate process, which means it is non-specific. Any bacteria that comes in, our phagocytes will do this. Dendritic cells are also phagocytes. Dendritic cells, we pointed out, was in our first line of defense in our skin and our mucous membranes. They are also phagocytic cells, so they are capable of this. And phagocytes will save a couple of pieces of the pathogen and then bring it by way of the lymphatic system to our adaptive immune response and have our B cells and T cells check it out. So that'll come back up too. So remember that is also going on in this process. All right, so that is phagocytic killing. I will also post this video so that you can watch that as well, because it's nice to see things in action. But not all microorganisms are capable of being phagocytized. What if you are being attacked by parasitic worms? So you've got worms in your system which are much bigger than cells, and your cells can't eat them up, can't phagocytize them. So some of these cells have non-phagocytic ways of killing. Eosinophils, remember these are the ones that come up with worms and allergies. Instead of phagocytizing these worms, which are much larger than them, they will actually bind to them and secrete some toxins. Interesting thing about this is that mitochondrial DNA of the eosinophils are involved. And that's kind of interesting because we don't think about DNA being a structure that does things. DNA is our instruction manual, right? It tells us how to do stuff. It doesn't actually do anything. But in this case, the mitochondrial DNA is involved in this process. So that's kind of an interesting thing. So that's a non-phagocytic way that some of these cells can attack microorganisms. We mentioned natural killer lymphocytes. We said most of the lymphocytes are going to be in those non-specific, that third line of defense. Most of our lymphocytes are going to be over here, but there were a few lymphocytes that were in the cells, the second line of defense, and these are those natural killer lymphocytes. They will also target cells and bind to cells and then kill those cells. Natural killer lymphocytes are designed to seek out cells that have been infected by a pathogen. So a lot of these are viruses. A virus comes in and infects the cells, and we've learned a lot about viruses. We know that once a cell has a virus in it, it's going to die either shortly or after a while, but that cell is marked for death. Natural killer lymphocytes, it's their job to find these cells and kill them before they can make a whole bunch of virions and spread that throughout the body. The other thing natural killer lymphocytes will do is they will target tumors. Tumors are our own cells. This is cancer, right? These are our own body cells that have lost their ability to regulate their cell replication, and they should not replicate, but some things get turned back on and they start replicating and make this mass, this ball of cells that becomes a tumor, and that can become a problem. It can take up space, or it can break off and cells can move into other areas of your body. And so this is cancer. We have our own defenses against cancer. Natural killer lymphocytes are designed to find those cells that are not behaving right, and they start making different proteins than they should, and that's how our natural killer cells find them. Our lymphocytes, natural killer lymphocytes find them. They will bind to them and destroy them. So when you hear things about a tumor was on the thing, a small tumor, and then it disappeared, our body does have defenses against this. Unfortunately, sometimes these cells are replicating much faster than our body can get rid of them, and that's where we have issues with cancer. So that is a type of non-phagocytic killing. Neutrophils are also able of producing these chemicals that will kill in a non-phagocytic way. All right, so go through phagocytosis. That is a big deal. Most of these cells kill by phagocytosis. Make sure you understand that, and then know these couple of different non-phagocytic ways of killing. Okay, so we are moving after cells, and the next video will take us to some chemicals in our second line of defense. Thanks, guys.